Hi folks, this is chapter 19 and we're not going to talk about everything in this chapter. In fact, we're going to cut out a lot of material on protein synthesis because that falls under the realm of genetics, so we'll be skipping that. The only thing we're going to look at here is some post-translational processing, so that'll be this short video, and then we'll look at some protein targeting and sorting. So this will kind of harken back to a discussion of the endomembrane system, but we'll be looking at how you get proteins targeted to places like the mitochondria and the chloroplast, so a little bit different. And then we'll look at a key technique because we've not done that for a little while. But this chapter's got a kind of important technique, and that is how you can study protein localization using fluorescently tagged proteins. So we'll do that at the end. So let's get going on our first section, which is post-translational processing. So after you've done translation, so that is after polypeptides are synthesized, they often have to undergo processes that are called collectively post-translational modification, and that's often a prerequisite for them to perform their normal cellular functions. So for example, in bacteria, the N-terminal methionine, which is N-formyl methionine, has to be removed, and in eukaryotes the methionine at the N-terminus is also removed. So that's two examples. What else? Sometimes you will find whole blocks of amino acids being removed from the polypeptide. For example, certain enzymes are synthesized as zymogens, and that generally causes them to be inactive, and then when they're activated by the removal of certain protein sequences, and that's a good example of that is digestive enzymes, where when they're synthesized in one tissue or one structure and then they're released into the digestive tract, you don't want them actually having the digestive activities where they're made. You only want them to have digestive activities where they're needed, and so they're produced as zymogens, and then they are post-translationally cleaved to be activated. And like you know that the transport of some proteins may also require the removal of certain sequences so they can be transported appropriately. And we'll talk about a variation of that when we talk about signal sequences in protein targeting and sorting in the next section. Let's look at one kind of somewhat complicated example of post-translational processing, and that is the example of insulin production. So the two subunits of mature insulin, which you see at the bottom down here, they come from a single precursor molecule called pre-proinsulin. And so the first thing that happens to pre-proinsulin is this N-terminal set of pre-amino acids is cleaved off. And then as what happens is these disulfide bonds get formed between what will be the mature A chain and the mature B chain. So if you look at it in the mature form, you've got a chain A and a chain B, and that's the mature molecule. And the two chains are linked together by these disulfide bonds. And so those disulfide bonds form here between within proinsulin to connect what will be mature chain A and mature chain B. But connecting chain A and chain B is this sequence of amino acids here, and that has to be removed from the proinsulin to produce the mature insulin. So that's a lot of post-translational cleavage of the amino acids, and that's kind of a little bit like splicing. In fact, there's a number of proteins which are produced who have sequences called inteins, which are similar to introns in immature mRNA, and they get spliced out, and then the other amino acid sequences get kind of glued back together, and those are called exteins. They're much like the exons in mature mRNA. So that's how you make insulin, with a lot of kind of post-translational cleavage modification. Other post-translational modifications might include chemical modifications of amino acids, such as phosphorylation, which we've talked about, and we'll talk about more in cell signaling, methylation of proteins, and also acetylation. So that's modification of the protein post-translationally, which is required for the appropriate either folding or enzymatic activity, or dimerization and formation of multimeric subunit structures. The only other one that really comes to mind is this kind of form of splicing, which I mentioned just now, where you remove inteins and link back together the exteins. So that's a little bit about post-translational processing. We'll leave it there, and then we'll get into protein targeting and sorting in our next video. I'll see you soon.